Okay, so welcome to this video. In this video what we're going to discuss is local anaesthetics. Okay, so the topic for this video is local anaesthetics. Now before we can discuss local anaesthetics and the mechanisms by which local anaesthetics actually work, what we need to discuss firstly is nociceptors, and we also need to discuss voltage-gated sodium channels, because the local anaesthetic drugs are going to work to block nociception by interfering with voltage-gated sodium channels and thereby blocking conduction of action potentials. And it's preferentially for neurons that are firing at a too rapid rate, which nociceptors are guilty of, basically, which is why you get a preference for blocking painful signal transduction without blocking all sensory modalities, and importantly without blocking motor neurons as well. Okay, right. So let's start off then by discussing nociception. Okay, so we're going to start off with basic concepts within nociception. So we're going to start off with the difference between a nociceptor and a nocicensor. Okay, so a nociceptor means the neuron which is capable of detecting painful stimuli. Okay, so let's just draw this out. So if we have a neuron here, basically it's a primary somatosensory neuron, so it will have a pseudo-unipolar structure, like so. Okay, so what does that mean when I say pseudo-unipolar? Well, basically here is the cell body with the nucleus in it here, and then you have a single axon coming off the cell body. Okay, so this is what is meant by pseudo-unipolar. Unipolar means that you have a single axon coming off the cell body. Now, why is this called pseudo-unipolar rather than just unipolar? Well, it's because later the axon actually divides into two portions. Okay, and it's as though this neuron is bipolar and has two axons. These two axons have completely separate functions. So that's why it's called pseudo-unipolar, because really functionally it is bipolar, but anatomically it actually is unipolar, which is why it's sort of half unipolar, half bipolar. Okay, so one of these axons is called the peripheral axon, and that will go off and have its terminal within some peripheral tissue, okay, potentially the skin or some visceral tissue. Okay, so this will go off to the peripheral tissue, and it's called the peripheral axon. Okay, whilst the other axon will go into the central nervous system, and this is called the central axon, so this will go into the spinal cord. So a nociceptor is a neuron which can be activated by painful stimuli. Okay, so painful stimuli such as, for instance, too hot conditions, too cold conditions, mechanically painful conditions. Okay, so too harsh pressure. In addition, certain chemicals such as too high proton concentration or ATP, which is indicative of cell damage. All of these chemicals would, all of these stimuli would classify as a painful stimuli. Okay, so the fancy term rather for painful stimuli is to call them noxious stimuli. Okay, so that's just a little bit of fancy terminology there for painful stimuli. So these noxious stimuli will activate the peripheral axon of the nociceptor, and it will cause an action potential to propagate up the nociceptor. Then it will propagate down the central axon, and it will be passed on to the spinal cord. Okay, so that's what a nociceptor is. It's the neuron which actually senses the noxious stimulus and fires an action potential, whereas the nocicensor is the actual receptor which the nociceptor, the neuron, has within it, which can detect the painful stimulus. Okay, so in the axon terminal of our nociceptor here, you will have some sort of receptor which can detect the noxious stimulus. Okay, so for instance, if your noxious stimulus is too hot conditions, then there will be some protein within the membrane of the axon terminal of this nociceptor which will respond to the too high temperature conditions and will cause an action potential to fire up this nociceptor. Okay, so often people think that the nociceptor means the receptor which actually detects the noxious stimulus because of the fact that you've got this septor here which is analogous to the septor in receptor, but don't let that confuse you. The nociceptor is the neuron. The nocicensor means the actual receptor that the neuron has within it for the noxious stimulus, whatever it may be. Okay, right. So that's the difference between a nociceptor and a nocicensor. Let's now talk about the difference between nociception, okay, and also pain. So nociception means the transduction of a noxious stimulus into an action potential which fires up a nociceptive neuron. Okay, so transduction just means taking one thing and turning it into another thing. Okay, so here we have the noxious stimulus and we are going to turn that into an action potential that will fire up the nociceptive neuron. That process is called transduction. Okay, so the transduction will be brought about by the nociceptor, which detects the noxious stimulus and then triggers the action potential. Okay, that process is called nociception. So the process whereby a noxious stimulus is turned into an action potential firing up a nociceptive neuron, that's known as nociception. Whereas pain means the conscious perception of something being horribly wrong. It means that unpleasant conscious sensation, basically. So pain is on the level of the brain, basically. This is generated by the entire brain, and it's not understood at all because we don't understand what the neural correlate of consciousness is. Okay? Whereas nociception is on the level of a single neuron, namely this nociceptive neuron, which it has its axon terminal within the periphery and is being acted upon by these noxious stimuli. Okay, so nociception we do understand, at least to some extent, whereas pain we don't understand really at all. We don't understand how you actually create that conscious sensation of unpleasantness. And obviously nociception is linked to pain. Often if the nociceptive neurons fire, that will trigger then this generation of this conscious unpleasant sensation. But it's not always the case that this one directly causes this one. For instance, you can have nociceptive neurons firing and then no conscious unpleasant sensations and no pain. Alternatively, you can also have pain when you don't actually have the nociceptions, you don't actually have the nociceptive neurons firing. Okay, right. So let's discuss these nociceptive neurons in a little bit more detail then. Okay, so all primary somatosensory neurons can be divided into 4 categories. Okay, so firstly, a primary somatosensory neuron means the first neuron which detects a sensory stimulus and is then going to relay an action potential to the central nervous system. So basically the neuron which actually detects the stimulus within the periphery then relays the information into the central nervous system. It's the same neuron, it's a single neuron that does the entire thing, and these neurons which do this are called primary somatosensory neurons. Now clearly not all primary somatosensory neurons are actually nociceptors, okay, so a subpopulation of the primary somatosensory neurons are then nociceptors, which are primary somatosensory neurons which respond to noxious stimuli, and when they activate the central nervous system, they promote the production of this pain, this conscious unpleasant sensation. Okay, right. So, all primary somatosensory neurons can be divided into 4 categories. Okay, and this division is done on the basis of their anatomy or their microanatomy, their histology, if you like. So basically, if you go to a cutaneous nerve, okay, let's say the radial nerve or something along those lines, and you cut it down the middle. So let's say here is the nerve. What you're going to do is cut it down the middle, take a cross section, and then have a look down the microscope at this little cross section. Basically, what you will then have done is you will have cut through all of the axons, okay? So basically, I've discussed that these nociceptors have this pseudo-unipolar structure here, and that's actually true of all primary somatosensory neurons. Their cell bodies are located very close to the spinal cord, okay, so those are far, far away. What we're currently looking at is the axon that extends between the terminals, which are out at the periphery, okay, and can be sensing the sensory stimulus, whether it's a painful sensory stimulus or some other form of sensory stimulus, okay, and then these axons are going to relay this information up to the point where we have the single axon which comes off the cell body, okay, bifurcating into these two axons, and then it will propagate through the other axon, which will take you into the central nervous system. Okay, and as I say, this bifurcation here, where the peripheral axon then becomes the central axon here, that will be very near the spinal cord, and we'll see that in a moment. So if we cut through a peripheral nerve, basically, we're going to be cutting through our axon here. Okay, so when we do this, what we'll see is our axons all in cross-section. And basically you see axons with different diameters, basically, and this is how axons of primary somatosensory neurons are classified, basically. Okay, and how we classify primary somatosensory neurons, we use the diameter of their axon and certain other characteristics of their axon, for instance, whether they're myelinated or not, to classify them into these four categories. Okay, right. So, basically these four categories are called A alpha, A beta, A delta, and then finally also C fibers. So let's talk about what each of these fibers' axons will look like if you cut through them in cross-section. So basically, A alpha are the giants, okay, so they have massive great axon diameters. So the diameter of an A alpha primary somatosensory neuron is generally around 20 micrometers, okay, and they are myelinated, so they do have a myelin sheath around them. So in red here I'll denote the myelin sheath, and they have quite a thick myelin sheath as well, so I'll thicken it up a little bit, okay. And this is going to result in these A alpha fibers being able to conduct action potentials very fast, because between the nodes of Ranvier you will get the conduction of the action potential via just diffusion of the depolarizing current, basically, from one node of Ranvier to the other. So basically, you don't have to propagate the action potential along the membrane underneath that cluster of myelin sheaths. So remember, an axon will look like this. Okay, you'll have clusters of myelin sheath like so, and then you'll have these little gaps in between these clusters of myelin sheath here. Okay, and these little gaps in between are then called the nodes of Ranvier. And the idea is that basically the action potential at this node of Ranvier can just jump to an action potential propagating along this node of Ranvier, which will then jump across this piece of myelin sheath here to this node of Ranvier. It doesn't have to propagate all along this membrane by saltatory conduction, which would take longer, basically. Instead, what can happen is, when you get an action potential here, the sodium ions that come in can travel all the way through here and will depolarize the membrane over here and then trigger an action potential here. Rather than it just being able to diffuse to the neighbouring portion and then causing an action potential there, sodium ions coming in here, causing an action potential in the neighbouring portion, etc. Okay, right. So that's A alpha fibres. A beta then are slightly smaller than A alpha. They're around half the size. Okay, so the diameter of an A beta fibre is then 10 micrometers. Okay, and A beta fibers are again myelinated. Okay, so they too have quite fast speeds of conduction, close to A alpha. Then we'll have A delta, and A delta gets smaller again, so their diameters are getting closer now to 5 micrometers. So this diameter is supposed to be 5 micrometers. And again, they are myelinated, so they will have a myelin sheath around their cell membrane. Okay, then we go down to C fibers. C fibers are not myelinated and they have very small diameter. So the diameter of a C fiber is only 1 micrometer. So this is the diameter of a C fiber. Now, C fibers cluster together within peripheral nerves. So basically, if we did do this cross-section of a peripheral nerve and we had a look at the C fibers within this cross-section, we find that the C fibers were all clustered together and they are within a Schwann cell. So they do have a Schwann cell looking after them. It's just the Schwann cell doesn't bother to produce any myelin to surround them, so they're unmyelinated. But they are surrounded by a peripheral Schwann cell which looks after them. Okay, but the Schwann cell doesn't just have one C fiber running through it, it will have multiple C fibers running through it. And the collection of all of these C fibers that run within this one Schwann cell, so I'm colouring now the Schwann cell in turquoise here. Okay, so this is the cytoplasm of the Schwann cell here. Okay, and you've got the C fibers running through it. Okay, so all of these are C fibers. 1, 2, 3, 4, 5, 6, 7, 8, 9. All 9 of them there. You call this collection of C fibers which run through this Schwann cell, you call them a fascicle basically. So this is a C fiber fascicle. Okay, right. So C fibers have the smallest diameter and they are unmyelinated. That's the key difference between these C fibers and the A fibers. Okay, right, so you have all of these different categories of primary somatosensory neurons. So you have a huge range of different size primary somatosensory neurons, basically. Very small ones, and then ones which are 20 times bigger, basically. So the question then becomes, the nociceptive primary somatosensory neurons, those which are detecting noxious stimuli and firing action potentials in response to those noxious stimuli, which are then being passed on to the central nervous system, which of these fiber types can be nociceptive neurons, or rather, the nociceptive neurons are which of these fiber types? I mean, they could be all. You could have nociceptive neurons which are all of them. Okay, so you could have nociceptors which are C fibers, and then other nociceptors which are A alpha fibers, but you don't generally find nociceptors which are A alpha fibers. Instead, you can, at a push, find nociceptors that are A beta fibers, A delta fibers, and C fibers. Okay, so nociceptive primary somatosensory neurons generally fall into one of these three categories. Now really, there are very few nociceptors which are actually A beta fibers. The main two that you find are A delta fibers and C fibers. Okay, so most nociceptive neurons will either be an A delta fiber or a C fiber. Now, the key way that we can therefore distinguish nociceptors into two groups is whether they are myelinated and therefore they are an A fiber or a C fiber. So that's generally where we go now. So what we do is we divide the nociceptors therefore into two groups. So there are the nociceptors which are myelinated, and those are the A fibers. Okay, so these are the myelinated nociceptors. Okay, and they're the A fiber nociceptors, so they'll either be A beta or A delta. And then there are the unmyelinated nociceptors, which will be C fibers. Okay, right. So there is some functional significance to the two different types of nociceptive neuron, basically. Okay? Basically, not only are the A fiber nociceptors larger and also myelinated, they also have much smaller receptive fields. Okay, and I'll talk about this just as soon as I've finished writing down this. So unmyelinated nociceptors are C fibers. So let's now talk about the fact that these A fibers have smaller receptive fields than the C fibers. Okay, so let's for a moment just say that these nociceptors are cutaneous nociceptors. So they have their peripheral processes within the skin. Okay, so let's just draw an easy picture of the skin. So here we have the epidermis, the outer layer here. Then we have the dermis. Okay, so let's say this is our nociceptor here, and these are its endings, basically. So what happens is once the peripheral axon here, okay, so this is the peripheral axon that has come from that bifurcation of the single axon which comes off the cell body. Okay, and then what happens is it starts to branch out into these multiple different branches like so. Okay, and these different branches are the terminals of this nociceptor. So at the end of each of these branches you'll have a little terminal which will have the nocicensors in, which can respond to the noxious stimuli, basically. Okay, so let's say this one that I've drawn here is a C fiber. Okay, and C fibers have larger receptive fields. Now what does this mean? This means that this branching at the end, where we form this like tree structure, will cover a larger area, basically. Okay, so if you think about it, if we have a noxious stimuli in any portion of this pink box that I've drawn here, this nociceptor will fire an action potential in response to that, because it's got axon terminals all over the place, and they will pick up any sort of noxious stimulus which is within this pink box, okay? And therefore this nociceptor will fire an action potential. Therefore, if we are receiving an action potential arriving in the central nervous system from this nociceptor, we do not know where it has actually come from within this box. It could have come from anywhere within this box. So, the information which comes from C fibers doesn't give you the ability to pinpoint very well your site of the noxious stimulus, Okay, so this pink box, which is the total sort of volume where the nociceptor is actually sensitive to noxious stimuli, this is known as the receptive field of that nociceptor. Okay, so C fibers have very broad receptive fields, whereas the A fibers generally have much more localized trees, basically. Okay, so they have much smaller receptive fields. So if I now draw an A fiber here, its little terminals might just cover a little area like so. Okay, so this then would be the receptive field of our A fiber here, this little pink box here. And therefore, if we're receiving an action potential from this A fiber nociceptor, we know much better where that noxious stimulus is actually present. So information coming in via the A fibers gives us a much greater ability to pinpoint exactly where the noxious stimulus is, basically. Okay, so the receptive field for A fibers is much smaller than the receptive field for C fibers. Now, what's the significance of this? Well, basically, it is a general principle, and it's not quite true, but it's a general principle that the A fibers mediate first pain. Okay, so in humans, basically, you will be familiar to the fact that we have two sensations of pain. Okay, so when you initially injure yourself, let's say, okay, so let's say someone punches you. Initially, it will hurt really, really badly. You'll get a very, very sharp form of pain. Okay, and that's because these A fibers will be firing. Okay, and when you've had that first injury and the A fibers are firing, you can pinpoint exactly where the pain is coming from. And that's because these A fibers firing, they tell you exactly where the noxious stimulus is. So let's imagine someone can deliver a really tiny punch to you. You'd be able to localize, or maybe let's say a pinprick, you'd be able to localize where that was, where that pain was coming from, because initially it's the A fibers which are firing, and they have much smaller receptive fields than the C fibers, and therefore when they do fire, you know that the noxious stimulus is coming from this smaller volume, and you can therefore better pinpoint where the source of the pain is. Whereas, well, that's known as first pain, is the first thing to say, that sharp initial pain. Whereas there is a second phase of pain known as second pain, which is the dull aching that you feel for a long time after the initial injury. Okay, so this is a dull, long, achy pain. Okay, now this is generally much more difficult to pinpoint exactly where the problem is, so it feels much more diffuse. Okay, and the reason for that is believed to be that it's mediated by these C fibers rather than the A fibers. And when the C fibers fire, you can't really tell nearly as well exactly where that noxious stimulus is present, basically. You've got a much larger region where it could be present. Okay, so you get much less detailed information, so the brain then gives you a much more diffuse pain, basically. You can't localize it. Okay, because the brain doesn't know where the pain is actually coming from as well as it did when A fibers were firing in response to the noxious stimulus. Okay, so that's a sort of oversimplification, but it's a helpful oversimplification. Okay, right. So let's now talk then about the transduction of painful stimuli then. So I want to discuss how a noxious stimulus actually induces an action potential within the neuron. And I'm going to take this also as an opportunity to discuss the action potential, because discussion of the action potential is going to pave the way for our discussion in depth of the voltage-gated sodium channels, because by looking at the action potential we can motivate our discussion of the voltage-gated sodium channels, since we'll see the properties that we need to explain basically. Okay, right, so I'll call it there for this video, and in the next video what we'll begin looking at is how a noxious stimulus, and the example we'll take is too hot, basically, extreme heat. Okay, so noxious heat. And we want to see how this is going to induce first a receptor potential in the axon terminal, and then an action potential in the axon of the nociceptor. Okay.